I am Nicole Letourneau, professor in the Faculty of Nursing and Cummings School of Medicine in Pediatric Psychiatry and Community Health Sciences. I'm also principal investigator of APrON and here to talk to you now about gene by environment interaction studies with APrON data. Back in 2008-2009, the APrON team had the forethought to collect DNA samples from children's blood or cheek cells when they were 3 months of age. I'm really grateful for that forethought, as the studies I will speak to you about now would not have been possible. These studies would also not have been possible without the help and guidance of many people. I want to especially thank Dr. de Koning and Dr. Palufo-Silveira, as well as Dr. Kobor and his incredible postdoctoral fellows, Dr. Moore and Dr. Merrill. Thank you so much for helping to advance this work. In a short 20 minutes, I'm going to speak to you about 5 exemplar projects examining gene by environment interactions with APrON data. 2 address genetic differential susceptibility, including a paper that's been published and a follow-up paper in preparation. 3 address epigenetics, with 2 focused on mothers' and fathers' exposure to adverse childhood experiences when they were children, and how that trauma or adversity impacts their baby's gene expression. The last paper in epigenetics examines how mothers' experiences of distress, for example, depression and anxiety during pregnancy, affects infants' immune cell gene expression. First, I'll focus on studies of genetic differential susceptibility. In both the studies of genetic differential susceptibility, we focus our attention on how parenting interacts with genes in predicting preschoolers' behavioral problems, such as attention or aggression issues, or how parenting interacts with polygenic risk scores in predicting preschoolers' security of attachment to their caregivers. These are important questions because both problems with preschoolers' behavior and insecure or disorganized attachment to parents predict an increased risk of mental health problems over the children's lifespans. So let's talk about the concept of genetic differential susceptibility first. Differential susceptibility has its roots in the biological sensitivity to context theory of Boyce and Ellis, and implies that a given genotype may result in a plastic phenotype, and thus a given individual may be placed along a continuum of genetic plasticity. According to this theory, genetic plasticity is primarily mediated by individual differences in neurobiological traits, such as a highly responsive stress physiology, in the case of the hypothalamic pituitary adrenal axis, which produces different phenotypic outcomes in either stressful or protective environments. In harsh environments, genetic plasticity may decrease individual ability to respond to threats and dangers, while in protective environments it may maximize ability to benefit from resources and support. In other words, the same genes that may be associated with maladaptive outcomes in negative environments may also provide more beneficial outcomes in positive environments. This differential susceptibility to environmental influences helps explain in part why not all children exposed to adverse child-rearing environments develop behavioral problems. My systematic review, and those of others, identified several genetic plasticity alleles implicated in behavioral problems in children and youth responding to stressful environments. These include polymorphic variants associated with dopamine receptors, DR, D2, and D4, dopamine active transporter 1, that is the DAT1, and the 5-hydroxytryptamine linked polymorphic region, or 5-HTT-LPR, monoamine oxidase A, or MAOA, brain-derived neurotrophic factor, or BDNF, and the cannabinoid receptor 1 gene, or CNR1. Jay Belsky, one of our newest APrON collaborators from UC Davis, created a plasticity index from these allelic variants. He included DAT1 10 repeat, DRD2, DRD4 7 repeat, 5-HTT-LPR, the short allele, MAOA 2 repeat and 3 repeat alleles. For each person, a score can be given to characterize whether the person has none of the alleles to all of the alleles. The more alleles a person has, the more the person is likely to be differentially susceptible to the environment. Belsky has used his index in several studies, and in one study shown here, he found for high school students with a greater number of plasticity alleles, the more well-adjusted they were under higher quality parenting conditions, and the less well-adjusted they were under lower quality parenting conditions. However, for youth with none or one plasticity allele, parenting quality did not matter. This is differential susceptibility. Now, let's talk more about parenting. That's the environment in the gene by environment studies I'm speaking about. In early life, parent-child relationships help regulate the child's response to stress, before the child is physiologically able to self-regulate. In other words, children develop their ability to regulate their stress response and behavior through their relationship with their maternal caregivers, especially during the first 3 years of life. Not surprisingly, it has long been established that low-quality parent-child relationships portend behavioral problems in children, while high-quality parent-child relationships are considered protective and may buffer the impacts of stress on child development. 3 main components constitute parent-child relationship quality: maternal sensitivity, control, and responsiveness. Healthy parent-child relationships are therefore typically characterized as sensitive and responsive interactions, attentive to infant needs, while mitigating excessive intrusion and control of the parent in the infant's activities or child's activities. Further, high maternal sensitivity contrasts with behaviors that are overtly or covertly hostile or excessively controlling. Interventions to support parenting and children's behavior may be compromised by a failure to consider differential susceptibility to experiences and the quality of the environment. So we sought to determine the interaction between parent-child relationship and biomarkers of differential susceptibility and children's behavioral problems in a normal sample. We also hypothesized that higher scores on the Plasticity Index would interact with measures of parent-child relationship quality in predicting better or worse outcomes, that is, children's behavior, in a differentially susceptible manner. We also hypothesized that individual allelic variants would also interact with parent-child relationship qualities in a differentially susceptible manner in predicting children's behavior. Our index was an adapted version of Belsky's index, as it included additional genetic differential susceptibility loci than Belsky, including an additional DAT1 loci, as well as BDNF and CNR1. Here they are for anyone interested in this later on in the conference video playback. And here is what we found when we looked at internalizing behaviors in preschoolers, that is, anxiety, depression, or withdrawal from social interaction. Higher scores on the Plasticity Index interacted with higher parental unresponsiveness in predicting more behavioral problems, and higher scores on the Plasticity Index interacted with lower parental unresponsiveness in predicting fewer problem behaviors, as indicated in the far right panel. Having an average Plasticity Index score did not interact with parenting quality at all, as indicated in the far left panel. Then we looked at externalizing behaviors, which include things like aggression and hyperactivity in the preschoolers. When we drilled down, we found that the CNR1A, or cannabinoid receptor gene loci, and the DAT19 repeat loci interacted with parenting quality in predicting children's aggression and hyperactivity. Specifically, the CNR1A plasticity allele interacted with higher parental controlling behaviors in predicting more problem behaviors. As well, CNR1 plasticity allele interacted with lower parental controlling behaviors in predicting fewer problem behaviors. The DAT19R had a similar pattern in that it interacted with higher parental controlling behaviors in predicting more problem behaviors, and the DAT19R interacted with lower parental controlling behaviors in predicting fewer behavioral problems, as you can see in the right side panels. However, when the alleles were absent, parenting had little impact on whether children presented as aggressive or hyperactive. Overall, we found evidence for genetic differential susceptibility to parenting in both internalizing and externalizing behaviors of 24-month-old children. To my knowledge, these findings are the first to come from a prospective cohort with strong measurement of parenting via observation. Past studies relied on retrospective self-report of youth or adults. Next, we found a terrific opportunity to work with Patricia Palupo-Silveira to examine genetic differential susceptibility using expression-based polygenic risk scores. Because we found that the CNR1A gene was such a strong plasticity allele, we focused on it next. Using expression-based polygenic risk scores, or EPRS, we can examine how the gene is expressed in the brain and how that interacts with the environment. For the CNR1 EPRS, our approach considers gene expression in the prefrontal cortex. So we sought to determine whether the EPRS for the CNR1 gene that we just showed interacted with parenting in predicting externalizing behaviors would interact with parenting again in predicting children's development of disorganized attachment pattern with their parent. The disorganized attachment pattern between a parent and preschool child is linked to psychopathology over the lifespan. So what did we find? For CNR1 expressed in the prefrontal cortex as a polygenic risk score, we found that either low or medium levels of CNR1 EPRS, high maternal sensitivity, reduced the probability of disorganized attachment, while low maternal sensitivity increased the probability of disorganized attachment. High CNR1 EPRS predicted relatively the same probability of disorganization at every level of maternal sensitivity. We also found that at either low or medium levels of CNR1 EPRS, high maternal controlling behavior increased the probability of child-parent disorganization, while low maternal controlling decreased the probability of disorganization. High CNR1 EPRS, in contrast, predicted relatively the same probability of disorganization at every level of maternal controlling behavior. We are preparing these findings for publication now, and next I will speak briefly about 3 epigenetic studies. So now I'm going to focus on the 3 epigenetic studies we have completed. But first, let me speak a little bit about epigenetics, especially social epigenetics. The next 2 slides are courtesy of my collaborators Mike Kobar and Sarah Merrill at the Center for Molecular Medicine and Therapeutics at UBC. Epigenetics are persistent, heritable alterations in genomic information that do not involve changes in DNA sequence. Therefore, while your genetic sequence never changes throughout your life, the resulting phenotypes can potentially change through epigenetic mechanisms. An epigenetic alteration in childhood could still be present in adulthood. Social epigenetics is based on the theory that experiences can affect various biological pathways, which can in turn influence epigenetic pathways such as DNA methylation, and potentially how our genes are expressed. While the genotype you're born with doesn't change, it does give you a window of potential expression that may be turned up or down based on epigenetic mechanisms. The specific mechanism we'll be talking about today is DNA methylation. DNA methylation is a relatively stable molecular tag on your genome that can act as a dimmer switch on a gene's expression, for example. DNA methylation is influenced by the environment. All 3 studies here may be conceived of as examining intergenerational environmental impacts of mothers' or fathers' experiences on their children's DNA methylation. Essentially, we are examining environments that may be prenatal or even preconception to see if they affect children's gene expression. And here's what we found in the first analysis. We measured fathers' exposure to adverse childhood experiences and DNA methylation of their offspring. Adverse childhood experiences include physical, emotional, and sexual abuse, emotional and physical neglect, and household dysfunction, including parental mental illness, violence, divorce, incarceration, and substance abuse. We found several genes in 3-month-old children were differentially methylated with increased paternal or father's childhood adversity experiences. They are depicted in red and blue at the top of the volcano plot. Here we examine the top 8 DNA methylation sites or loci identified in the volcano plot by undertaking a gene ontology or GO analysis. Warmer colors indicate greater significance. The top few indicate associations with meiotic chromosome segregation, chloride channel, and transmembrane transport activity in the 3-month-old children. These relate to cell function and transcription regulation and metabolic functions. Intriguingly, one of the top genes was related to circadian rhythms. Then we examined the top 8 DNA methylation sites or loci identified in the volcano plot to see what outcomes they associate with in children. Positive correlations are depicted here in red and negative correlations in blue, with bolder colors indicating stronger associations. You can then see here that children's attention and hyperactivity at 3 years of age is negatively associated with 2 loci on the right of the diagram. One loci in the middle associates positively with infant sleep latency at 3 months of age. Sleep latency is defined as longer time to fall asleep, which is an indicator of infant sleep problems. In the next paper to be submitted soon, we examined how mother's exposure to adverse childhood experiences impacts her 3-month-old's DNA methylation. Analyses considered perinatal distress as an important covariate, such as depression during pregnancy, along with maternal cortisol as a measure of prenatal stress. The figure on the right here shows significant results in both infant blood and buccal cells correlated methylated regions, or CMRs. Overall, we did not find that associations between maternal ACEs and DNA methylation were attributable to perinatal distress levels in either tissue, that is blood or buccal cells of children. I quote here from the paper, "In terms of competing explanations, we find better evidence for possible epigenetic patterns passed from mother to infant independent of the in utero environmental experiences of distress or stress. It is also possible that early life, however, context in the children's histories, such as their parenting, shaped infant DNA methylation early in postnatal life, which could not be ruled out with the DNA methylation quantified so early at 3 months of age." For this study, further, we did not explore the genes for the loci to see what behaviors they link to, but that will be a next step. However, when we examine maternal prenatal distress variables alone, that is depression, pregnancy-specific anxiety, and recent stressful life events, not considering mother's history of adverse childhood experiences, we found something intriguing. This analysis was led by former postdoctoral fellow and now APrON investigator Dr. Kara Ross. Here you can see coefficients in the bottom of the middle column of the table that provide evidence that late pregnancy-specific anxiety, early and late pregnancy depression, and late pregnancy stressful life events affected 3-month-olds' gene expression, and particularly in relation to genes associated with immune signaling. Taken together, these 3 studies suggest that perhaps ACEs exert unique influences on children's DNA methylation in an intergenerational way, as do prenatal distress variables. In summary, we found clear evidence for differential susceptibility to the parental caregiving environment in the APrON cohort. Our latest work, in preparation with Dr. Palufo Silveira, is offering a more sophisticated approach to examining differential susceptibility with EPRS scores for the CNR1, and Patricia recently received a large CIHR grant to examine gene-by-environment interactions in APrON and other cohorts in greater depth. With respect to epigenetics, we found evidence for intergenerational transmission of impacts of parenting and stress during pregnancy on next-generation outcomes. Next, we will examine nutrient impacts, for example, vitamin D, on infant DNA methylation with Mike Cobor and Yvonne Lemers, and interactions with neurotoxins with Deborah Dewey and other Australian colleagues. I would also like to examine the impacts of ACEs on mothers and fathers themselves. Thank you so much for listening to me today. I'm happy to take a couple of questions in the time we have left. You can also find out more on APrON's website or reach out to me by email. My published books provide a lot more background on genetics and epigenetics and impacts on child health and development. Thank you again.